Good morning, everyone. My name is Yujia Zhang, and I'm a PhD student in Shanghai Jiaotong University. And I have been an exchange student in the University of Tokyo in the past 1 year. So today, I'm excited to present what I have done in the past 1 year in Nakai Senses Lab. That is a tool for predicting tumor evolution pattern by inferring spatial copy number variation distributions. Firstly, I want to briefly introduce the background. Tumor evolution or carcinogenesis is a multi-step process, including the morphologic, genetic, and epigenetic changes. Here is an example of gastric cancer. We can see that from the normal gastric tissue to the cancer tissue, it will undertake several stages, including the chronic atrophic gastritis, the intestinal metaplasia, the dysplasia, and finally the gastric cancer. So that raised our interest in exploring the changes in this kind of process. But what can we do? For a long time, histopathology has been the gold standard for tumor diagnosis and prognosis because it can provide us detailed information under the microscope. But the images alone cannot provide the molecular feature. So if we want to do a more comprehensive diagnosis, we have to make a combination with the molecular diagnostics. So in recent years, we've seen significant advancements in pathology. In 19th century, we only have cellular pathology, that is H&E and IHC images that can only separate the patients into two groups. And in 21st century, we have molecular pathology, and we have many more new technology like NGS, FISH, and PCR, which can provide us more information. And now we have spatial omics that make us to combine the spatial features with the DNA, RNA, and protein information, and can make a more comprehensive diagnosis. However, the challenge of studying tumor evolution here is how can we explore the tumor evolution from the snapshot data? Usually, we only got the static information like the spatial omics or single-cell RNA sequencing for one time point. And cannot capture the dynamic progression of the tumor over time. However, by applying some algorithm with the spatial omics framework, we can begin to infer the trajectory of the tumor evolution even from this kind of one-time point observations. And the main idea here we use is copy number variation calling, which is a feasible approach to explore the tumor evolution. CMV refers to a circumstance of the copy number variation of a DNA fragment and is strongly related to the cancer cell growth, drug sensitivity, and resistance. Also, in different types of cancer, the tumor clone diversity is different. So our model focuses on using the CMV data to explore and predict the tumor evolution. And there is some existing method to infer the CMV with single-cell data. The main idea is to use the smooth gene expression data to infer the genetic information from the gene expression. However, if we directly use this kind of tool on spatial omics, it cannot capture the spatial information because it doesn't consider the spatial relationships. So our model wants to develop a spatial temporal model to predict the tumor evolution by inferring spatial copy number variation distributions from spatial omics data. Here is our model called SCOIGET, that is spatial copy number inference by graph on evolution of tumor. And the main idea is from the spatial omics, we got the input of transcriptomes and spatial locations. By combining these two parts into the spatial graph, we construct the original spatial graph. And using the graph neural network, we can capture the feature from the graph and get the latent representation. And by the model decoder, we can reconstruct the spatial graph. And by the iterative training, we can make the model stronger. So about the data, here we use the two data sets of prostate cancer and colorectal cancer. And both data use the inferred CMV that didn't consider the spatial information in the original paper. And they are using 10x genomics with spatial omics with the whole genome sequencing method. And like other studies do, firstly, we use the data pre-processing and do some quality control on the spatial omics data. And then we do the gene annotation and binding. It is that we annotate the genes based on their chromosome positions and group the genes, like 25 genes, into one bin to reduce the data sparsity and noise, and especially reduce the data dimension because it is quite highly dimensioned data. And next step, we construct the spatial neighbor graph. That is to calculate the adjacency matrix and calculate the spatial statistics like Morris I to evaluate the spatial autocorrelation between stop. And here is our model training. We use the spatial neighbor graph and graph attention layers to learn and extract the important features in our model. And we optimize the model using the reconstruction loss MSE to enhance the performance. And here goes to our result part. Our model contains 3 modules, and the first module is copy number calculation. It is to infer the copy number variations by our model. This part is to estimate the copy number and deploy spot identification. These 2 parts, the first one is to identify the reliable deployed spots based on the self-correlation scores. And we make initial CMV estimates for each spot based on the result of our model. And then we determine the breakpoints of the copy number changes and make the copy number profiling. We identify the breakpoints using the PELT algorithm, which can detect the segmentation of the changes. And we generate the copy number profiles for each spot and spatial clone. The second module is the spatial domain identification, also from the latent, the result from latent representation. And here we construct the integrated spatial maps. We incorporate the genetic information learned from the module 1. It is the copy number variation result. And we add this kind of information into our model and refine the copy number estimates and produce the detailed spatial map. And we use some clustering method like lead-in. We can get the spatial map clustering of the tissue. And the third module is the inference of tumor evolution patterns, which is the main goal of our research, is to construct the phylogenetic tree of tumor clone. This part, we construct the evolutionary tree and of the tumor, use the learned embeddings, and we elucidate the tumor progression and heterogeneity. So in this part, we can begin to give some biological explanations on the tissue and to make the sequence of each clone based on their relationship. Here gives the summary of our research. Our model offers a novel framework for understanding tumor evolution through spatial omics data. It integrates multiple features, use graph neural networks to predict CMV patterns and tumor progression. And our model is highly adaptable to various tasks and various data types. Also, we need some further validation to fully explore the robustness across different cancer types. Finally, thank you for watching, and thanks for Nakai Sciences Lab and Professor Hui Lu's lab, and thanks for listening.
Yeah. So when you say spatial data, then you're talking about only the single cell, or DNA, and then RNA, and then protein?
The data we use is RNA sequencing, the spatial transcriptomes.
So you have taken both the copy number as well as the gene expression, or just the copy number?
We use gene expression to infer the copy number. So because only the expression data, they doesn't contain the genetic information, but as we use the expression data as a reference, and we can infer the.
You have mapped the loads.
Yeah.
Okay. Thank you.